Okay, we're going to start with Unit 1, PowerPoint 1, Part A. Again, I want to mention, please make sure you refer back to your outline regularly. Unit 1 and 2 are both very work-intensive because we are setting the foundation for the course, and then 3, 4, and 5 are a lot less work, time-intensive. So make sure you put that effort in the beginning, and then the second 3/4 of the class is going to be a lot simpler. So with the start of this class, I'm going to mention that hands are by far the most dangerous and important tool for anyone in the allied health or healthcare field. You don't put on a pair of gloves and decide you're going to save your employer in the hospital a lot of money and just keep the same ones on all the time. In fact, what you want to do is change them absolutely regularly, because as you can see on this picture, whether we're talking about influenza or RSV or Pseudomonas, everything sticks to your hands, and then you are passing that pathogen along as you go from patient to patient. So we are going to cover the scientists first. Leeuwenhoek is known as the father of microbiology. It took over a century or 100 years for people to duplicate what he first began with. He took water samples and fecal matter scrapings from teeth and started looking at them with something that looked like a prism or a magnifying glass in the sun. So he is actually known as the father of the microscope, or discovered it, which is a big deal because bacteria are tiny and you cannot see them without the use of a microscope. The next scientist, and I just want to give you the caveat, I do speak a little bit quicker, but you have these recorded, so it is simple for you to listen to them as much as you would like. So we have Francisco Redi. He is also one of the founders of microbiology, and he was first to dispute something known as spontaneous generation using a maggot experiment. Spontaneous generation is saying that life comes from non-life, and we all know that life comes from other living cells. So in this first particular jar, you see a piece of meat. The flies are coming into the jar, laying their eggs, and you see maggots developing. He wanted to know where that essentially came from. Now you have the second jar, and in this case, notice there is a cork or a stopper on top. So the flies are not actually able to enter the jar and lay their eggs. Do maggots develop? No, there's actually no maggots developing here. Now, if you look at the third jar, there is almost a mesh or a cheesecloth on top of it. So the flies may be able to lay their eggs on the mesh, but they're not actually able to get to the meat, and therefore no maggots actually develop or are grown. So this basically shows that life has to touch life. Living things have to come from living things. Maggots are not going to come if they're not able to touch that meat. So the explanation of spontaneous generation is something of life coming from non-living matter which does not exist. In fact, in science, we have biogenesis, and this is life coming from other pre-existing life. Interestingly enough, all the way up until the mid-1800s, people thought that it was juju or simply bad air or bad karma that caused infectious diseases. They didn't understand bacteria and viruses and protozoa and other types of microbes that cause illness and the concept of biogenesis. This term of thinking infectious diseases come from bad air is known as miasma. Next, we have to mention epidemiology. It's an incredibly important field, and now with the current state of affairs going on, and specifically pandemics, this is studying health issues, and you want to look at all the W's. Who, what, when, where, how, why, what is the source, or what was patient zero? What actually caused this health issue? And the mode of transmission is usually going to be one of three things. It's either airborne, waterborne, or foodborne. So how does this pass and become communicable between person and person? The father of epidemiology, or one of them, there are two. The first one is Semmelweis. What he actually discovered is a way to stop maternal and newborn deaths from being quite so high. It might sound silly, but back in the day, someone would be in the barn and they would be taking care of animals, and then they would walk into a bedroom and deliver a newborn baby. It never really occurred to them to actually wash their hands. So at one point he realized something as simple as washing hands, and he used a chlorinated lime. He didn't quite have Clorox bleach and Clorox wipes, And essentially what happened, just simply by washing hands and not transferring the source or the pathogen from one place to the mother, because birthing is a bloody process, the maternal death rate as well as the newborn rate instantly decreased. The second father of epidemiology is John Snow. This happened in London. So if you take a look at this map over here on the right, notice all of these little dots. They're representative of people that died in this particular area. And then in the middle, there is a red circle and a little X. So people were dying of some unknown illness. They couldn't figure out the source or the transmission of how it was passing from person to person. And eventually it was figured out that all of the people, even though they were all these dots in various areas throughout different socioeconomic classes, just completely different neighborhoods, were all walking to this one particular water pump. Think about filling buckets of water to take it back to your home. This is known as the Broad Street Pump, and it was a huge outbreak of cholera. He was the father of epidemiology in the sense that he determined all of these people in this vast area of London, the common source was this one particular water pump. To this day, it is actually memorialized on Broad Street, and this statue or thing you're looking at in the picture does exist. So this is known as the first official epidemiological study. The next scientist is Pasteur. You might think of pasteurizing milk because this is where it comes from. And he also proved biogenesis, which is again, life arises from other life. And he disproved spontaneous generation. And this is the experiment at how he managed to do that. These are known as swan neck flasks. He has a media, which is filled with all kinds of nutrients, and it's bubbling. This would make sense to all of us because after a hurricane or an earthquake or tornado, any type of natural disaster, the first thing you're told to do is boil your water to sterilize it, to get all of the components and bacteria and everything out of it. So in this case, he's boiling this infusion, and notice the steam is going to escape from this swan neck flasks. This thing sits for a while, and absolutely no microbial growth appears. And you know that because the liquid in here, or the infusion, is nice and clear. There is air moving in and out of this particular flask. Several months pass, and there still is no contamination. So at this point, it's understood that it's sterile. But if you look at the bottom of the swan neck flask, there is dirt and dust and other things that have settled in it, but it didn't have the gravity to make its way up this swan neck. Next, he has another sterile infusion because ahead of time it was boiled, which is what essentially makes it sterile. And at some point, this swan neck flask was tilted and put on its side. In a matter of hours, microbes started to appear and it was contaminated, which essentially shows that biogenesis, life comes from life, and that you need to have some type of contact of nutrients and the shifting of air and everything else in order for the contamination or microbes to reappear. So later he figured out the concept of pasteurization, which is what we do with milk, and the concept there is to reduce the number of microbes. Once something is open, it never is perfectly sterile, but a certain number of microbes is always okay. He came up with the germ theory of disease, which basically showed and explained that microorganisms are responsible for the cause of diseases. So it is life from life, and he proves biogenesis and disproves spontaneous generation. Next, there's Koch. He then went ahead and took Pasteur's work and he verified the germ theory of disease. He basically looked at colonies that were grown on a solid surface. He added something known as agar or media that turned it into a gelatin, and then some of them were liquid, which were other pure cultures. If something is pure or a pure culture, that implies that there's only one type of organism growing in it. If we were to lick a petri plate and culture your mouth, it would absolutely not be pure because our mouths are incredibly dirty. Whereas if something is done in a sterile lab environment, it is just one type, so it's known as a pure culture. You need to be familiar with Koch's postulates. You are absolutely going to know and understand this germ theory because he did in fact verify it. So here is his experiment. So here's Koch's experiment. It's simplified in 4 steps, but we're going to talk about it for a moment. The purpose of this is to verify the source of an infection, or the source of the illness, or the source of the demise where someone becomes ill or actually passes away from the illness. This was done using rabbits. So there was a rabbit that died. Right here, this is part A. And they pulled some blood, spun down the plasma, and then cultured it. Here are the individual colonies. Notice you're seeing 3 different colors. There's red, purple, and black, which implies that 3 different types of organisms were isolated from this infectious animal. So now the question is, which of those 3 different type of organisms actually caused the animal to die? Next portion was to take healthy, susceptible animals, and notice the 3 colors. There's the red, the purple, and the dark bluish black. They were all injected into healthy, susceptible animals, and again, these are isolated organisms, 1 in each. After a certain incubation period, one of the animals, or one of the rabbits, died, and the other 2 were okay, which essentially tells you one of those 3 are the virulent or pathogenic organism. Then what happens is it needs to be isolated and pure cultures and plated. So if the question were which one caused the healthy, susceptible animal to die, you would essentially say whatever is representative of that red organism. There's a very important step between part C and part D, which you will be asked to explain at a later time. You cannot just do a test like this one single time. Most things in science and experiments are done in duplicate, if not even triplicate, so it is done multiple times. Again, you have an animal that became infected, and then you isolated those different organisms. You inject them individually into healthy, susceptible animals. One of them succumbs to the particular organism, then that one is plated. Now, if these happen to have 2 different colors in them, let's say this plate had not only the dark blue and black, but also the red, that's not isolated. So now the test would need to be done again. The purpose of Koch's postulates, again, is to determine which particular susceptible or suspected bacteria infected the animal in the first place. So if you think about it, there are many different organisms found all around us. CFU stands for colony forming units. This is a measure of the growth on a particular plate or in a broth tube if it's not gelatin, and a solid media type of plate. Notice in the soil there is the highest number of units found per milliliter. So they are also floating around in the air, in the water. Keep in mind that many pathogens are airborne, some are waterborne, and the last mode of transmission is foodborne. I just want to mention one more time to ensure that everybody remembers Unit 1 and 2 are incredibly involved, and then it calms down a little bit. So don't be discouraged. Make sure you watch these PowerPoints multiple times so that you're able to retain the information. And now take this and go back to the outline and fill in notes for you to be able to study.